For the library of integrated network-based cellular signatures, links, many gene expression signatures using the L1000 technology have been produced. The L1000 technology is a cost-effective method to measure gene expression in large scale. The Links Canvas browser, which I'm going to demo today, is an interactive HTML5 web-based software application that facilitates querying, browsing, and interrogating almost all the currently available Links L1000 data. The Links Canvas browser implements two compacted layered canvases that you can see here. One is used to visualize clustered L1000 expression data, and the other displays enrichment analysis using 30 different gene set libraries. Clicking on an experimental condition, highlight gene sets enriched for the differentially expressed gene from the selected experiment. There is also a search interface that allows users to input gene lists and query them against over 100,000 conditions. In this demo, I'm going to go over in detail describing each component of the Links Canvas browser. So we will start with the browser interface. As I said, the red canvas, the one that is on the left, visualizes the L1000 experiments. Each tile represents an experimental condition, and that experimental condition is actually representing 8 experiments, 4 controls, and 4 treatment conditions. The tile brightness tells you overall how different the treatment is from the control. So the perturbations that cause a significant change have brighter red. You can change the contrast to only highlight the most significant perturbations or see the overall picture of all of the experiments on this particular canvas. Each canvas represents all of the experiments that were done in a specific cell line in a specific batch. For example, the default batch is called LGP004 and it was applied to 6 breast cancer cell lines. The current selection is for the BT20 cell line. If I change that to MCF10A, now I'm looking at a completely new set of experiments from the same batch where different ligands were applied to stimulate MCF10A cells. Mouse hovering on each tile show you the details of each experiment. For example, this experiment profiled the effect of treating MCF10A cells with SCF at 3 nanomolars and measuring gene expression after 6 hours. This perturbation is the most significant perturbation on this particular canvas. The experimental canvas also had 3 other views. A time point view. Here you can see that there were 4 different time points: 1 hour, 6 hours, 8 hours, and 24 hours. The experiments seemed to be clustered based on the time point. The drug view shows you the various types of drugs or ligands. You can also see clustering by drug or ligand. For example, all of these pink perturbations are from IL-1 stimulation. And there is also a dose view. And in this particular batch, there were 6 doses, and you can see some clustering based on dose. So the experiments on the canvas are clustered using simulated annealing to try to place experimental results as close as possible based on their similarity, computed using the correlation coefficient of the L1000 differentially expressed gene vectors. If you want to highlight several experiments, you can use the multiple selection and then select those experiments. For example, here I'm interested in IL-1. So I highlighted all the IL-1 that belong to that specific cluster, and then that selection can remain as I toggle between the different views. So the IL-1 experiments always stay at their specific location, and the different details about the perturbation are shown in the different views. You can also notice that the canvas folds on itself. So this top edge is connected with the bottom edge, and the sides are also connected. Once you click on a tile, enrichment analysis is displayed on the enrichment analysis canvas. This canvas is visualizing a gene set library. In a gene set library, each term is associated with a list of genes. We're looking at overlap between the upregulated genes that were induced by CSF at 3 nanomolars measured at 6 hours in MCF10A cells. These are the upregulated genes, and we're looking for overlap between those genes and genes that, when you knock them out in mice, produce a specific phenotype. For example, one of those terms in this specific gene set library that was created from the MGI mammalian phenotype ontology, the lists on the enrichment canvas are also organized based on similarity. So the bright cyan areas are areas where lists have many similar genes. There are also different views in the enrichment analysis canvases. You have the grid view, the p-value view, which only highlights those enriched terms. The lower the p-value, the brighter the tile. There is also a network view where the enriched terms are connected. You can click here is opening up in a pop-up window, and you can arrange the enriched terms on this network and view all the labels of the enriched terms at once. Here you can see that there are 4 different clusters of enriched terms that share some gene lists. And finally, and probably the most useful, is a table view of rank enriched terms. And in this table view, you can see all the enriched terms, and when you mouse over, you can see the overlapping genes. So those genes overlap between the list of abnormal eye physiology genes as well as the list of upregulated genes upon SCF stimulation in MCF10A cells. You can also toggle to look at what happens to the down-regulated genes, and immediately we get those terms. The mammalian phenotype gene set library is only one of 30 gene set libraries that you can choose from. Those gene set libraries are divided into 5 categories: transcription, pathways, ontologies, diseases and drugs, cell types, and miscellaneous. The transcription category tries to identify the transcription factor, histone modification, and microRNAs that potentially regulate the differentially expressed genes. For example, we process the ENCODE dataset to identify gene lists from over 600 experiments performed by the ENCODE project. Selecting the ENCODE gene set library immediately switches the enrichment analysis for ENCODE, identifying GTF2B in K562 cells as the most enriched transcription factor. So that means that the downregulated genes after CSF stimulation are most likely regulated by the transcription factor GTF2B as measured in K562 cells. And this is one experiment from the ENCODE project. And these are the genes that overlap between the downregulated genes and the putative target for GTF2B as they were identified by ChIP-seq. The upregulated genes appear to be potentially regulated by CTF-CF. So this means that CSF is potentially upstream of CTCF. The browser interface also provides a search capability. In this search box, you can identify perturbation for specific ligand in specific cell types at specific time points. For example, I'm interested in identifying the molecular mechanisms that are potentially affected by the small molecules that is found in red wine resveratrol. So there are many L1000 gene expression experiments that were applied using resveratrol. I will pick the first one that was applied at 24 hours. When resveratrol was applied at 10 micromolars and gene expression was measured at 24 hours, this compound did not cause a significant change compared to the other perturbations on the canvas. If I examine the enrichment analysis for the transcription factors, I can see that the most significant perturbation is for E2F4 in K562 cells. I can tell that because of the brightness of the white dot. If I switch to the table view, I can see all of the transcription factors, and we can see that there is 2 entries for E2F4 and 1 entry for E2F2 and 1 entry for E2F1 at the top of the list for the resveratrol perturbation. You can also see 2 entries for MAX. This suggests that those transcription factors are potentially downstream and are activated upon resveratrol stimulation of A375 cells. If we switch to the downregulated genes, we can see a strong signal for NF-YA, NF-kappa B, and IRF3. Also, CEBP beta is listed as one of the top transcription factors. And this is supported in the idea that resveratrol is a potential anti-inflammatory because NF-kappa B, IRF3, and NF-Y alpha are members of the immune system, potentially turning on an inflammatory response. We can now look at pathway enrichment analysis, and I'm choosing the KEGG pathways. And you can see that resveratrol downregulates the expression of genes that are associated with Parkinson's disease and other neurodegenerative diseases. It upregulates genes that are associated with pancreatic cancer, inositol phosphate metabolism, and chronic myeloid leukemia. When looking at the mouse phenotypes that are enriched for the genes that are activated by resveratrol, we can see that abnormal survival and mortality and aging are found to be enriched terms. It is suspected that resveratrol, through its activity on the enzyme SIRT, affects the aging process. But what we see also is that it potentially has a link to pathways and genes that are associated with cancer. One thing to keep in mind that we have to be very cautious about the conclusion that we are drawing from such analyses. The Links Canvas browser is an exploratory tool and should be used with caution. It can only be suggestive of potential effects of drugs and ligands on cells. So now I switch to the search interface. So this is the Links Canvas browser search engine. There are 2 input boxes. One is for up list of genes and the other one is for down list of genes. These are intended for users to enter their own up and down gene lists from their own experiments. If you click on the example, the software fills the boxes of the up and down genes for you. You have to select if you want to analyze only the up or both or only the down for the search. The idea is that we are going to identify experiments that are similar to the experiments that induce the changes in gene expression. So once we selected the up or down genes and input our list of genes from our own experiments, we can decide whether we want to find experiments that induce the same change or experiments that reverse the expression. The aggravate option should be used to identify similar compounds or ligands that induce a change that was observed experimentally by the user. The reverse option simply swap the up and down lists and search for perturbation that can reverse the expression. For example, trying to find compounds or ligands that can switch the expression to the control condition. And this potentially can be useful to identify drugs that can be used to treat diseases. If you're comparing normal tissue and diseased tissue, the program can suggest small molecules that can switch the expression of the diseased tissue towards the normal condition. Let's keep it at aggravate and perform the search. The search doesn't take more than 30 seconds. Now the program returned the top 50 matched perturbation to my input list. Clicking on the top perturbation takes me back to the browser interface. The search interface shows only the top 10 perturbation, but you can navigate to other pages by using the previous and next buttons. This particular example was taken from this perturbation. That's why we have very high overlap that almost sums up to 1. 1 is 100% overlap. So this number represents a normalized measure of overlap between my input list and the up and down gene list from the experiment. The help link provides you a link to the user manual and a video tutorial. This tool is still at the prototype stage, so we appreciate your input and feedback. Thank you for watching, and I hope you'll find this tool very useful.